In this lecture, I'm going to talk about what I call the carbon story of cellular respiration. This story starts with the process of glycolysis, stage number 1 of cellular respiration. If we look at this word, it's glycolysis. Glyco is referring to sugar. Lysis means break or breaking. So that's what happened. We're breaking a sugar. And what sugar is that? It is glucose. Glucose is made of 6 carbons. I'm going to draw them as these spheres here. They're actually in a little bit different form in the cell, but I don't care. I just want you to track the carbons. So there's 6 of them, and this is glucose. Let's mark that in teal because it's an input. And what are we going to do? We're breaking. Now here's some really key concepts. One key concept is that you break a bond, you release electrons. Overall, the point of this is to extract electrons. So we're going to see that we break a lot of bonds in order to do this. Glycolysis is happening in the cytoplasm of the cell. That's the part of the cell that is not any of the compartments. It's the kind of goo part, the interior. So what happens when we break the bond? We know we can extract electrons. So how are we going to do that? Here's another concept that's used over and over in this process. There is a molecule called Nad Plus. We can think of it as a cab or taxi that can carry electrons. When it's in the plus form, you can think of that as the light being on. So that's an empty cab. You've never hailed a cab because there's ride shares now. When it's empty, the light is on, so then you know you can flag it down to get in it because you know that nobody's in that cab. So this is the same thing. The light is on. It means an empty cab, which really what I'm meaning here is that it's no electrons inside. When it has electrons and picks them up, its passengers make it into NADH. So now this is the form where the light is off. You see there's no more plus. And that is indicating, that it's a full cab, as in the electrons are in it now. So who comes in? The empty cab with the light on, Nad+, picks up its electron passengers, and it leaves as NADH, a full cab. And we're going to see that over and over again, where does NADH take the electrons? They go to the ETC, or electron transport chain. There's a lecture about the electron transport chain that you can also watch. Often when we break a bond, we can also make some ATP. But just a little bit in this case. So that's an output. So let's mark it in this gold color. I'm going to draw it in a starburst because it's a high energy molecule. It's ultimately the goal of cellular respiration. But we're just making a little. Now some cells are small enough to where they can run just off of glycolysis, and it's enough ATP. But many cells actually need the cellular respiration process to occur, and glycolysis isn't enough. Or they can just run for a small amount of time using glycolysis. So what happens after we break the glucose in half? Well, we get 3 carbon compounds, well, we get 2 3-carbon compounds. And this molecule is called pyruvate or pyruvate. Okay, so now we have 2 pyruvates that have more electrons in their bonds we want to extract. So we're going to do what I call 2A, and in our book they call those the preparation reactions. They happen in a compartment inside the cell. So pyruvate is transported into that compartment, and we call that compartment the mitochondrion. And specifically, it's in the matrix of the mitochondrion, which is the interior space. I have a lecture about mitochondria, so you can watch that to get more information. So the pyruvate goes in, it's transported, and I'm going to just color this last carbon here so we can track it. The other pyruvate does the same thing, but I'm just going to do one of them. Realize that it happens twice. So we're going to break the bond to extract electrons. So we break off this last carbon, and it actually leaves as a CO2 molecule. CO2 is carbon dioxide. Let's mark that in the gold color to show that it's an output. It's just a waste product, so it diffuses out of the mitochondrion and out of the cell. And in humans, we breathe it out. That's where the CO2 that you breathe out comes from. And you'll notice that where did it come from? It came from the glucose, from the food. So it's a gas that you breathe out that starts out as food. So carbon dioxide does not come from oxygen. Carbon dioxide, yes, it's a gas, but that doesn't matter. It's the carbons from the food you eat that are broken off that are released as CO2. We broke a bond so we can extract some electrons. So who comes in? The empty cap company, Nad Plus, picks up those electrons and leaves as a full cab as NADH. Again, this is going to go to the electron transport chain. And then in order for this 2-carbon compound that we have now from breaking off the CO2 to be reactive, another molecule is added on called coenzyme A or CoA. So it's very large. And when it's added on, I'm draw a wiggly bond because it makes the molecule reactive and unstable. And I'll draw some lines around to show that it's an unstable reactive molecule. And this molecule is called acetyl-CoA or acetyl-CoA. And we'll remind ourselves that it's unstable and reactive. So that's good because it gets it to be able to go into the last part of the carbon story, what's called 2B, the citric acid cycle. Citric acid cycle starts with a 4-carbon compound, and that 4-carbon compound gets the 2 carbons added to it. So it bumps off the CoA, and then we have the 4 carbons, and then those 2 carbons that we just brought in attached. So it switches out basically the CoA for the 4-carbon compound and sticks those 2 on. And I'm going to be very general for the rest of the cycle. What do we want to do? We want to extract electrons. So how are we going to do that? We break off carbons in the form of, like this one, let's say, CO2. There's another place where we have our output. Again, it diffuses out of the cell. First the mitochondrion, and then the cell. And you'd breathe it out. We have this happen a few times. I don't care about that. I just need you to know generally what's going on. So after we break that bond, we know that we can extract electrons. So who comes in? Empty cab company, Nad Plus. Picks up its electrons, turns into the full cab, NADH. Guess where that goes? To the electron transport chain. In the citric acid cycle, we also have another cab company called FAD, which can pick up electrons as well and leaves as FADH2. That's just another full cab. This extraction happens a few times. And another part of the process is that a little bit of ATP can be made. Not enough to run the whole cell, but a little bit. So again, high energy molecule, the goal of cellular respiration, is also made in the citric acid cycle. And then the starter molecule, this 4-carbon compound, is regenerated. And that's why it's a cycle. So as you can see, we start with food, glucose, and we break off CO2s in order to release electrons and extract them from the bonds. Those CO2s are breathed out to the atmosphere. Those electrons are extracted and carried by NADH and FADH2 to the electron transport chain. We have 3 steps to the carbon story: glycolysis, preparation reactions, and the citric acid cycle. I have another lecture that talks about what happens at the electron transport chain, so you should definitely watch that as well.